Nakikita na niyo? Hello?
Yes po, Doc.
Okay, sorry na unstable dito ng Wi-Fi. So these are the individual conidial growth. Oh Lord, kaya Lord. Okay. Then we have the annelide. This involves elongation with production of each conidium and acquire the ring-shaped scars or annulations at the point of conidial production. So, because of the presence of these annulations, they are collectively referred to as the annelides. So, we have the exophyala and the scapulorhyosis. Yeah. So this one na lang. So because of these annulations, okay. So they are called as the annelides or annelidic conidiogenesis. Okay. So we need also to define some terms. When you say conidiophore, it is a specialized conidium bearing hypha. Just like in my template slides. Okay, so this is a conidiophore. Then we have the sporangium. These are the sac-like structure in which the asexual spores are produced. So the entire illustration did contemplate is the conidiophore. And then ito igbaw is referred to as the sporangium. And the sporangium is in turn made up of sporangiophores. So ini nga grupo, as a group, it is referred to as sporangium. Tapos individually, they are referred to as in sporangiophores. The individual pores. Okay? So, adin man, mas maka-appreciate nimo. So, sporangium, this is the collection of the sporangiophores. So, an individual na. Okay. Now, we'll go now to the sexual reproduction. So, just like in the eukaryotic cell, may the male and female sexes involved. So, this is initiated in cases of fungal organism with the production of plasmogamy. So, plasmogamy, this refers to the two compatible haploid nuclei that are together in the same cell. And then we have also the term karyogamy. This involves fusion of two nuclei to form now the diploid nucleus and may immediately follow plasmogamy or may be delayed. So, iton kuan pala, an individual palang nuclei, they are referred to as plasmogamy. Then, when they fuse together, then amo na itong term karyogamy. Did you understand? So, once they fuse na, mag-fuse na ang plasmogamy, mahimo na nga karyogamy. So, God. Yeah. Okay. So, when there's in case of sexual reproduction, so we have this life cycle of the fungal organism. So we have the haplophase, the dikaryophase, and the diplophase. So in haplophase, This involves unicellular or multicellular thallus that contains now the haploid nuclei. Then we have the dikaryophase. This involves two genetically distinct haploid nuclei that occupy each cell of the thallus. And then from the dikaryophase, it will now go or enter the diplophase, wherein the diploid nucleus formed as a result of the karyogamy. So from the union of karyogamy, it will now become as plasmogamy. Hey, there is also so-called parasexual reproduction. So this refers to the sequence of events that culminate in genetic exchange via mitotic recombination. So this is initiated by the formation of heterokaryon. So what is this parasexual reproduction? So the heterokaryon, the thallus containing haploid nuclei of two different genotypes. So what this heterokaryon is most commonly formed by hyphal anastomosis and nuclear exchange between genetically different strains of the same species. So itong parasexual reproduction, simply it is a result of anastomosis. Nagchlorosla ang duha ka hyphal cell, nag-anastomose tapos diretso na nag-exchange na hin genetic material. So sana paro-pareho gihapon nitong tao para waray commitment, kakaptan lang nimo tapos diretso na exchange of genetic materials. Sana all. Di ba? Uy, may dadidin nagkuan happy face. Namamati ngayon kamuha. Sana gihapon para tong waray mga label nga mga relationships, tapos may dala nimo crush, and then you are afraid of the commitments, magkakaputay la ka mo, tapos exchange dayon of genetic material. Okay? But diri la ininhiya kuan. Diri this is bisang patiya parasexual kung humans, it's between man and woman. Diri same na sexes. Di ba? Kuan Cesar. So, diri pwede kuan ini magkakaputay paro lalaki nga magbuburo ng usa. Diri. Okay. So, this is an example of heterokaryon. So, individual hyphal cell just anastomose each other and exchange genetic materials. Okay. So, how do the fungal infections produce? So, this involved establishment of mycotic infection that depends now on the size of the inoculum and resistance of the host. So, what is very important here is diri karusingon nga we are having may the opportunistic na infection, fungal agent na magkakamayada kadayon fungal infection. So it is dependent on the size of the inoculum. Size of the inoculum. Ang kadamo han fungal na spores na imo na ingest or the resistance of the host. So the severity of the infection depends mostly on the host response to the organism. Just like what I said earlier, that some of the infections are opportunistic in nature. So if you have good immune response, then sometimes you can evade from these infections. So what are the determinants of pathogenicity of the development of fungal infections? So Most common pathogenic fungi do not produce toxins. They do not produce toxins. However, many pathogenic fungi show physiologic modifications that enhance their ability to invade the tissues, like increasing the metabolic rate or modify the metabolic pathways or modify now the cell wall structures. So pathogenic fungi are thermotolerant. So meaning they can tolerate higher temperatures and resist the effects of the active oxygen radicals released during the respiratory burst of the phagocytes. So, okay. So, it is not through toxin production, but modifications in the metabolic system or pathways of the fungal organism that cause the disease. Okay, so what are the clinical manifestations of the disease of fungal infection? First is hypersensitivity reactions. Please take note that some fungal antigens are allergenic. So they are antigenic also. But I have told you earlier na most of the fungal organisms are non-toxin producer, but there are also some fungal organisms that cause fungal disease through toxins. So toxicosis, we have the so-called mycotoxicosis. This is brought about by ingestion of feeds and food products contaminated by toxin-producing fungi. So typical example of this is what? The aflatoxin that is contained in your peanuts. Mahilig. Sige lat kuan samsam hinton mani, waray kikita alamagon na ngay anan iya mani. Maupay kun an iba nga mani ang ginaalamang sensor. Then we have the mycetismus. This is caused by ingestion of fungi containing preformed toxins. So, may daluwat in preformed toxins like in mushroom poisoning, di ba? Pero ayaw luwat niyo i-generalize kaya may daluwat edible na mushroom. So, depende daluwat itong type of the mushroom. Okay. Then another is clinical presentation of fungal diseases is, of course, the development of infection. So these results from the direct invasion of tissues or organs. So the infections caused by fungal organisms are divided according to what part of the body is involved. So it may be superficial. So when you say superficial, it may involve only the cutaneous layer, namely your skin and some of the skin appendages. So they are referred to as superficial mucosis. They are confined to the outer layers of the skin, then the nail or hair. So the fungi that are involved are collectively referred to as dermatophytes. Then we have the cutaneous mycosis. These are confined to the subcutaneous layer of the skin and rarely spread systematically. So they are just confined to the subcutaneous layer. Diri hira nag-i-spread. Then the third group of fungal infections are referred to as systemic mycosis. This involve now the viscera and become now widely disseminated So fungi that cause systemic mycosis have specific predilection to various organs. So may daliwat niya predilection. Like for example, the aflatoxin, mostly they goes to your liver. Okay? So how do you diagnose fungal infections? So you can do your microscopic examination of patient samples. So you can do your KOH mount. So potassium hydroxide test. So this is the simple, fast way to detect dermatophytes. This is very simple. You just get kuan in blade na ano ton tapos i-scrape lang nimo itong sample tapos you add drop of potassium hydroxide and then examine immediately under the microscope. Then you can do serological tests like direct immunofluorescence. This is to identify the fungi in fixed tissue sections, mga biopsy na inihiya. Then you can do also staining like using your Gomori methenamine silver stain. So this stain the fungal cells black in tissue sections. Okay. Another is serological testing for fungal antigens. So you can have mga slide tests like your latex agglutination test. This is a typical example of this is for cryptococcal antigen detections. And of course, the gold standard, if you are dealing here with infection, is culture and isolation. Culture and isolation. So if during the board examination, whenever you are asked about isolation, how do you diagnose in mga bacterial, fungal, or viral infection, definitive diagnostic procedure is always culture and isolation. Of course, using with appropriate culture media. So the pathogenic fungi are usually grown in saboroutes, dextrose agar. So itong SDA, is the usual culture medium used for the fungal organism. And then you can add inhibitors. This is to make it more specific because like for example you can add cyclohexidine, penicillin, streptomycin, or other inhibitory antibiotic to prevent contamination of bacterial or saprophytic fungal organisms. So para maging specific itong saboroutes, dextrose agar for the isolation, may that niya mga added inhibitors. Like for example, your penicillin. What is the purpose of incorporating penicillin in SDA? This for the purpose of inhibiting the gram-negative organism. Positive organism. Gram-positive organism. Okay? So, unlike in bacterial culture that you have only one setup, that is incubating now your bacterial culture medium in the incubator set at 37 degrees centigrade. But if you are dealing here with fungal infection, for the fungal isolation and culture, you need two setups. One, incubated in the incubator set at 37 degrees centigrade, and one as in the environment lab. So in the room temperature of 25 degrees centigrade. This is for the purpose of demonstrating if the organism you are isolating has this dimorphism property. Okay, then under also culture and isolation, how do you now out and identify the isolated organism? You can describe it macroscopically through the using the following or describing the width of the mycelium, the presence of septation, branching, pigmentation. You can describe many colors of the colonies you will enjoy. Then the spore or the conidia production. So, macroscopic pala ito niya. And then, another one is to do your microscopic morphology description. So, under the microscope na here, then just like your bacterial organism, you can do your biochemical reactions, or to make it more specific in the identification, you can do your nucleic acid probing. Okay, so aside from culture and isolation, you can also do indirect methods based on the host immune response. Like you can do patch testing, you can do skin testing, then serological tests like latex agglutination test that detects now the presence of immunoglobulin M, which signifies recent infection, or you can do complement fixation test that detects now immunoglobulin G that tells us chronic infection. Okay, that's for the introduction.